All right, we're so glad to have Xinnan Wang here as our speaker today. She's actually with the Department of Neurosurgery at Stanford and is doing cutting edge research on Parkinson's disease. And she's going to speak with us today about the biomarker that she's exploring and how that relates to drug development for Parkinson's disease. And let me just indicate, she is a shareholder of a few companies, a co-founder, advisor, and shareholder of AccureX Inc. Did I pronounce that right, Xinnan?
Yeah.
Close enough, okay. And also the shareholder of Mitochondria Inc. And I'll send those out to everybody on the chat if people are interested. So, and Xinnan invites anybody to use the chat or the question and answer box as we go along, and she will monitor that, and I will certainly monitor that, and we'll go along and answer your questions. So take it away, Xinnan.
Thank you so much, Robin, for the introduction and also for having me here. Hello, everyone. I'm very glad to meet you all here virtually, and we can discuss some ongoing work in my lab about biomarker and drug development for Parkinson's disease. I will try my best to talk about research in a layman's language, but please feel free to stop me or interrupt if you have questions or you don't understand the word jargon I may say during my talk. So there's really no need for me to introduce Parkinson's disease to this crowd. Just a little history of Parkinson's disease. More than 200 years ago, a British neurologist, James Parkinson, first described a group of symptoms, later named after him this group of symptoms that people named as Parkinson's disease. He wrote an essay, this is called an essay on shaking palsy. So the first time he described symptoms of typical Parkinson's disease patients. And we all know that Parkinson's disease is a movement disorder. So if we have at least two of these four cardinal symptoms at the clinic, we could be diagnosed as Parkinson's disease, although there are many individual specific symptoms involving many other symptoms as well. Genetically, most of the PD cases are called sporadic, which means you don't really have any relatives you know who had the disease, or you may sequence your gene at 23andMe, and there is no mutation in the known PD causing genes. So that fraction is about 90 to 95% of total cases that we call sporadic. The rest is very small fraction. We call it familial. So familial, which means that you have relatives, sometimes very close relatives, who are known to have Parkinson's disease or Parkinson's-like symptoms, or you sequence your genes and you found, there's mutations in well-known Parkinson's disease causing genes. So we call those cases familial. But those cases really represent a tiny fraction of the total cases. For some really rare cases, I don't know if you know, for example, these two genes called PIN1 and the Parkin. Mutations in these two genes, recessive means the two copies of this gene are mutated. That's called recessive. So people who had those mutations will have the symptoms very early in their 20s. As you know, most of the PD patients had their symptoms late in life, at least 50, 60 years old. So this very rare case, genetic case, we call it early onset. So a famous example of this early onset disease patient is Michael J. Fox, a famous advocate and also an actor for Parkinson's disease. And he is an early onset case. but it is publicly unknown what genes that he may have mutated in his gene. So this is some basics of Parkinson's disease, and you probably know all of this already. We also all know that there are quite a few unmet needs in Parkinson's disease field. First of all, you'll be surprised that there's a high rate of misdiagnosis in clinic. People have estimated that there are about 10 to 20% of cases that are misdiagnosed. So if you are diagnosed as Parkinson's disease, or we diagnose 10 Parkinson's patients, maybe 2 of them, they don't really have Parkinson's. So why is that? Because the symptoms, especially in the early stage, look very similar to a couple of other movement disorders, such as essential tremor, PSP, MSA, CBD, in case you're familiar with these terms. So it is really hard to distinguish. And then we also know that the sole criteria right now to diagnose a Parkinson's disease patient rely on neurological evaluation. So there is no molecular marker to definitively confirm this person has Parkinson's disease. So this marker I will talk about later, and then this is the key to both diagnosis and the drug development. But currently there's no reliable marker in this case for assisting.
Will you be speaking later, Robin, will you be speaking later as to a definition of a biomarker and maybe some examples in other diseases?
Yes, I will elaborate later.
Perfect.
And then we can all understand what is a biomarker. But here I just want to emphasize how important it is. And there's no reliable biomarker for now for Parkinson's disease. So without this marker, about 20% of the patients are misdiagnosed. And also that will hinder the development of drugs. I will elaborate that later too. So here just, we know there are a lot of obstacles and all kind of winding down to a molecular marker that will really help us to solve these problems. So our lab really hope to solve some of these challenges. Clearly, these challenges limit moving therapies from the lab to hospitals, to patients. Okay, so before I talk about biomarker, I really want to also emphasize that our lab is trying to find a cure for Parkinson's disease. But before we get into too deep about this topic, I really want to emphasize again that a cure relies on addressing the cause of the neuron cell death rather than the symptom. So why is this so important? Because we all probably know that Parkinson's disease is caused by death of a particular neuron in your midbrain called dopamine producing neurons. So the death of neurons are very progressive. When a patient gets the symptom, there's about 50% of neurons already died, but the dying process is continuing. So it doesn't stop. In the current symptom relieving management, a lot of them, especially the new innovative ones, are very effective. We see very striking relief of symptoms, such as deep brain stimulation. There's also new stimulation or brain stimulation techniques that can relieve the symptoms. However, these really cool techniques rely on a live neuron in our brain. So if the neurons keep dying, those management will eventually lose their effect and then will eventually cannot be effective anymore. Therefore, our goal in my lab is to find out what really caused the neurons to die, and then can we find a way to stop neurons from dying, rather than just simply relieve the symptoms by stimulating the remaining neurons. So we think that this is the key to find a cure. We need to know how neurons die. So then we can stop this process. We can address why neurons die, and that's the only way to find a cure. So our lab has a very compelling hypothesis that we think it could be one of the reasons neurons die in Parkinson's disease patients. Before I talk about that, I'd like to introduce a jargon here. It's called mitochondria. Mitochondria are, this word is a Greek word. It's a two Greek word put together, mito and chondria. Both means the shape of this organelle in the cell. So here is a beautiful picture of a mitochondrion. Mitochondria are bean-like, thread-like, elongated. So that is what mito and chondria means in Greek. In addition to how beautiful it is, so what does mitochondria do? To make the best analogy, let's say our cell is a factory or a city. Every single protein, every single organelle is a person or a machinery or a device or a car or a restaurant. So everything in the city is performed by a protein or an organelle. Imagine that how we run our city. We need energy, right? We need, like humans need to eat, and our buildings need electricity, and our cars need gas. So all these are energy. We need energy to power every single activity of our city. This is exactly the same for a cell as well. The cell needs energy to power all the protein activity, all the ribosome activity, all the organelles, and then power this movement and the surviving, resisting to stressors. And this energy are all produced by mitochondria. So then you can see how important mitochondria are to the cell. So those are the energy plant. We call it energy plant to a cell. And this is the best picture I found online. And you can see this is exactly like this is mitochondria that producing all the energy to everybody in this city. Another picture also very interesting. If we make another analogy, the cell is this factory, and every single protein has a part, but mitochondria are the most important in this case because they produce all the energy a cell needs. So without mitochondria, cells cannot survive. We cannot survive. So this is how important these organelles are to our neurons and cells. Next, I will introduce another jargon, another term, I would say. So let's say mitochondria are the power plants for the cell. And when mitochondria are functional, we call it healthy mitochondria. So that just the factory, the plant is very functional. Therefore, they can produce all the energy this city needs. But during the aging of our cells, mitochondria can be damaged. There are many reasons, many factors that can damage mitochondria, especially aging. So when we age, there are a lot of factors associated with aging that can damage mitochondria. So imagine the power plants are not working, and then we call these power plants damaged mitochondria. This is a very important definition in Parkinson's disease. When mitochondria are damaged, you can imagine that the cell cannot have sufficient energy because mitochondria are the power plants to the cell. And you may feel fatigue. Sometimes a lot of patients feel fatigue, and that's really because your mitochondria are not producing energy that needed to support the activities. But in addition to that, more importantly, the damaged mitochondria emit toxins to the cell. Make another analogy, like we could imagine that a used car, very old car, it will not pass smoke test, and then it will emit pollutions into the air. So that's just like damaged mitochondria. But what damaged mitochondria emit to the cell, the rest of the cell, are very, very toxic, even more toxic than the pollution from a used car. Because that toxin, damaged mitochondria release, can kill the cells easily. So how would a healthy cell to try to avoid damaged mitochondrial accumulation because of this release of toxins to the cell that will kill the cell? Then I will introduce another term, two Greek words put together called mitophagy. Phagy means eating and mito means mitochondria. So mitophagy means eating the mitochondria. The easy way to explain what mitophagy is, I would say that it's a car towing system in a cell. If we have used a car that's not functional and emitting all these toxins to the rest of the cell, then we need to have a car towing system to identify those cars and then go take them out in order to avoid the release of those toxins to the cell. That process is called mitophagy. Healthy cells have a very efficient mitophagy process. Once there is a damaged mitochondria in the cell, the cell can quickly spot that mitochondria, distinguish it from the rest of the healthy cars, functional cars, and then it quickly go there to take this used car, releasing pollution into the air, to quickly take it out and then recycle, eliminate the further damage to the rest of the cell. So mitophagy, this car towing system, is crucial to healthy cell, to make the cell healthy, to avoid the damage, further damage from this bad used mitochondria. So our hypothesis for the cause of Parkinson's disease is that this car towing system is broken down. Therefore, a lot of used cars are accumulated in our neurons. And the consequences, first, there's no energy being produced because these cars are not working. These power plants are not producing energy. And then second, most importantly, that they emit this pollution into the air. It's so bad, it will kill the neurons eventually. So we think that this could be the cause. And we have very strong evidence in animal models and cells to show that the mitophagy process, the car towing system, is really broken down, causing the cell death. And the cell death is the reason in patients, they exhibit the symptoms, the movement, the motor symptoms, and other symptoms as well. So if this is true, how we are going to prevent the cause or address the cause. A little further information about the work in our lab is that we found a single protein called Miro1, and this protein is on the mitochondria. So this protein is on the surface of any car, all the cars in the city. And we discovered that Miro1 has to be removed from the used car in order for the car towing system to identify the used car and then take them out. So Miro need to be gone in order for the car towing system to work. In Parkinson's disease patients, removing Miro is impaired. So then you can imagine that Miro is not being removed. Miro stays on used car. So the car towing system is not going to recognize those used car. Therefore, there will be an accumulation of those used car that they keep emitting toxins to the cell. They're supposed to be taken out from by the car towing system, by the mitophagy process. But because of Miro is there, Miro is not being removed in Parkinson's disease patient. Therefore, the damaged mitochondria cannot be cleared, eventually leading to accumulation of toxins and eventually cell death. So before I talk about how we are going to address that, I want to show you some real data So those are, the red is kind of, you can imagine those are mirror. Those are the protein levels of mirror. And each individual column is a person. And those are our controls. They're either healthy or people with other diseases. And then Parkinson's disease patients are here. And then we also have a few Parkinson's at-risk individuals. And we have a big population in this case. We have almost 74 individuals that either have Parkinson's or at risk for Parkinson's disease. At risk means they have the known Parkinson's mutations, or they're the relatives, younger family members of the Parkinson's disease patient. So they're at high risk to develop this disease. So now you can see that we damage mitochondria. So the first row are healthy, and the second row, we damage mitochondria. And then for this control group, the color switch from red to black means mirror is gone, mirror is removed. And this is good. This is good for mitophagy because mitophagy can occur. So the cartoon system can come and then take out those damaged mitochondria. But you can see how striking it is. And these are some statistic analysis. So there is a lack of color change in most of the PD patients, very striking. So mirror cannot be removed, and that will affect the following cartoon system to recognize those damaged mitochondria and eventually causing neuron death. So how did we do that? So here we get skin cells from Parkinson's patients or control subjects by a simple biopsy. And a lot of you already did this, I believe, in your arm, either forearm. I think most of it is in the forearm. And then we can culture the cells, which is called fibroblast, skin fibroblast. We can culture these cells in the lab, and then we can detect mirror here using biochemical assays. So we can test mirror in a tissue very accessible to us, just from your arm. And it's minimally invasive. You almost feel no pain. And then we can detect a molecular defect. This is pretty amazing to us because we don't have to get to the neurons of a patient or the brain of a patient, which probably will really invasive, you know. So the reason is this is very exciting is that I will bring in another term called biomarker. So here is where the biomarker stands. If we are able to detect a molecule, a protein, and then a defect of this protein in a tissue that we can easily guide, for example, skin cells or a blood cells that we do it regularly for everybody when we go to hospitals, without damaging, without getting into people's brain or spinal cord or any other procedure that's very, very painful or invasive. And then if we can do that, like we just did for mirror, then this method really has a potential as to serve as a biomarker. So biomarker, which really just means a marker for the disease progression or initiation, and we can mark different stages by looking at the alteration of this protein. And this has to be significantly associated with most of the patients. So therefore, we know this is not only for certain individuals, but represent the majority of the patients. So there are twofold of significance of our study here, that we can detect a striking association of mirror1 defect with Parkinson's disease and the risk using skin fibroblast of patients. First, I mentioned about misdiagnosis. Because of the symptom similarity between Parkinson's disease with a lot of other diseases. So if we have this assay, in addition to the neurological evaluation, we are able to double confirm whether this patient really has Parkinson's disease. So this is significant because when we have the treatment are different for different diseases. So a confirmation of the diagnosis is crucial for therapeutic intervention. And the second fold of significance of a biomarker is for drug development. So without a biomarker, it is really hard to develop drugs, develop treatment, just because we can't really see how the drug works. So here is really a jargon. It's called a pharmacodynamic marker for target engagement. This really means that if you have a drug, you really want to have a marker to monitor how the drug is effective, especially in clinical trials, when you give this drug to a patient. In the case of MIR1 marker as a biomarker, I will use this picture to explain how biomarker works. So imagine that we have a potential patient that may display some PD-like symptoms at the clinic. We don't really know if he or she has Parkinson's disease. So we could just extract their skin fibroblast by a simple skin biopsy and culture the cells in the lab and then test the assay that I just showed you in the previous slide. We test whether this individual is positive or negative for a MIR1 defect. So this is the step one. We test MIR1 marker using their cells. If it's a positive, then we can test the compounds that can eliminate MIR1 marker. Here we call it MIR1 reducer because in Parkinson's disease patient, MIR1 is not being removed. So we want to reduce MIR1. So without giving those compounds directly to this individual, we first test in a culture dish in a lab, but using their cells to see if MIR1 reducer can really eliminate this marker, or in another word, can really rescue the phenotype or eliminate the defect of MIR1. So if it's effective, then we give this compound to this individual. And then we can continue to monitor, so doing it again to get the cells after a couple months, doing it again to see if the MIR1 defect is really eliminated after we gave the drug to this person after he or she takes into his body. So in this case, you can see in every single step, we have a marker. We have a way to know first whether this person is positive for MIR1, and second, whether those cells have a defect in MIR1, and then whether the compound will be useful for this person. And then lastly, we still have a marker to, after the person takes the compound for some time, we can see if the MIR1 defect is really eliminated by the compound. And then we can combine these, we can correlate with the MIR1 defect with the movement, the symptom relief from the patient to see if the molecular defect can be eliminated. In the meantime, the patient's symptoms can be improved. So this is what we call coupling a potential diagnostic tool or marker with MIR1-based drug development. Why this is so important? Because currently, all the clinical trial, none of them has any marker. So you can imagine that if you don't have any marker, you simply test any compounds to the individual, you're basically blind. You have no idea how the drug would affect any protein. And if the drug is not effective, does that mean the drug is not strong enough or it does not target the right target? So it is crucial to have a marker in future clinical trials. And that may be why current trials all failed, because there's no marker for the compounds they're testing. Okay, so what we are now.
I had a couple questions.
Sure, yeah.
Related to this slide and what you just said. You mentioned that there can be up to 20% of people diagnosed with Parkinson's disease who actually don't have Parkinson's disease. They might have one of these atypical Parkinsonism disorders like PSP. Mm-hmm. Do we know that people with these atypical Parkinsonism disorders don't have this same MIRO1 defect?
Yeah, so that's a really good question. We did test a couple patients. We have a couple PSP patients and a couple CBD patients, and they are negative for the MIRO1 marker. But the limitation, so the good news is our marker can really distinguish real Parkinson's disease patient from other Parkinson's-like patients because the treatment will be different. But we have the limitation of our current study is we only have a few patients due to the insufficient donors. In this case, we got a lot of Parkinson's donors, but not a lot, for example, from PSP, which is totally different pathology from Parkinson's disease. So the current data show that these few patients, they are negative for the MIRO1 marker. But I think larger population is needed in this case to have a definitive answer.
And how about for multiple system atrophy? Do you have any of those in that pool?
Yeah, that's such a good question. We didn't get any MSA patient. That's the disease name, right? The short name. Right. Yeah, so unfortunately we didn't get any patient, but I believe Stanford movement disorder, they enrolled some patients that in the future there could be some fibroblasts available to us that we can test the MIRO1 marker in those patients.
Okay, thank you.
But that's a really good question because we want to know like whether like in a sense that if those patients do show MIRO1 marker, that's also important because we can treat these patients maybe similarly with our compounds, right?
Absolutely.
So I think either way would be really, really important to know. Right?
Yeah, this is a little bit of a tangent, but are you familiar with SYN1? And SYN is SYN for synuclein 1. Oh, yeah, it's I'll call it an experimental skin test used to diagnose, quote unquote, diagnose Parkinson's disease. And a lot of lay people have been in our support group and elsewhere have been asking me about this SYN1 test recently because there are some doctors who are saying that they are offering this test as a diagnosis for Parkinson's disease. Could you explain a little bit about why that test which is not FDA approved yet as a diagnostic tool for Parkinson's disease, but the approach in a way seems very similar in terms of testing to the approach you're taking?
Yeah, so that is a very promising approach as well. It works very differently mechanistically. So I call it alpha-synuclein. So alpha-synuclein is a protein. We know that about 99% of PD patients in their brain, they have this pathology called the Lewy body. And the major protein composition in Lewy body is this protein called alpha-synuclein. So it accumulates in the brain, even in healthy aging individuals, but that is a pathological hallmark for Parkinson's disease patient. It's so high as 99% almost. So Lewy body only occur in the brain, in neurons, in the nerve cells. And the way that the new biomarker assay you described is that recent study, people have discovered that this protein can also localize to nerves. They start from the brain, but they can also either they originated from the peripheral or they move from the brain to the peripheral. So they can be present in the nerve terminals in your skin. So that's how it works. And imagine if that's really 99% of PD patients have alpha-synuclein accumulation in the neurons, then the assay should work really effectively, right? So basically how it works is different from this biopsy. So this biopsy only takes cells and then we culture it later. So the biopsy that they're doing is they will also take portion of your neurons. So our skin cells, there's a neural terminals in our skin. So they will need to take some of the nerve and then they can stain the protein. They can see if this individual has alpha-synuclein in those nerves. So theoretically, it sounds very promising, but there are different reports regarding how effective, how accurate this assay is currently for Parkinson's disease diagnosis. And I believe people are trying to get FDA approval at this stage. I think that's just as far as I know, but I think we'll need more research for either assay to confirm in larger scale patient, in more larger population of patient to confirm the positive correlation of the marker and the disease diagnosis.
Right. And one other question comes to mind is, could you explain a little bit about biomarkers for other diseases, like a biomarker for diabetes or other diseases, just to give people an appreciation of how powerful a biomarker would be?
Yeah, so I think a very good example is probably high lipid. I think diabetes is another good example, but I don't know the word for that. So if we have a high lipid in our blood, that will cause hypertension, for example. So when you go to clinic and get your blood test, you always see the different molecules in your blood test, right? Like LDL, some numbers, right?
Cholesterol.
Cholesterol, exactly. So there's a good and bad cholesterol, and those are biomarkers as well, in a way. So if and the similar for this mirror test. So if certain molecule exceeds the normal range for a big healthy population, for example LDL, then you have a high chance to develop high cholesterol related diseases such as hypertension, diabetes. So that's just one example that we all know what is a biomarker. So I think in a sense biomarker is more complicated than that, and then it requires really multiple groups to confirm your molecular changes. Like for Miro, it really marks either the start of the disease or the progression of the disease. Yeah, let me think about other examples.
No, that's good. Thank you very much. Yeah.
Yeah. Yeah. I think for regular blood draw, there are a lot of molecules that potentially serve as a biomarker, right? So that's basically the idea that if some markers are not normal, not within the normal range, then we need to think about the possibility of a disease. That's how it is. But I think a key thing is the biomarker need to be easily, so the tissue that you detect biomarker need to be easily accessible, like blood draw or skin biopsy. So currently we are also working on a test on Miro from a blood test, a blood draw. I think in the future that could be more convenient for patient if we can do that as well. Okay. So where we are right now, we propose this couple a marker with drug development, and then we think this strategy will really improve the success of future clinical trials. And as you can see, we have tested about 100 patients, their fibroblast we have tested neuron marker, and then we discover about 94% of the PD patients show positive neuron defect. We have tested several of them with a compound we developed that target the neuron directly, and all of the patients, the compound is effective for the cells from all the patients we tested so far. It's about 6 patients so far. So you can see we are about here. The next step is to give the compound to a real person, a real patient, rather than the cells. So this step requires FDA approval. So we're at here right now. And the FDA needs a lot of data, needs a lot of proof concept data to show safety, efficacy, and the reliability of our compound, and also some population study as well. So we are here. But while we are trying to guide to the compound to the clinical trial, I can tell you a little bit about this compound. So this compound we discovered through an artificial intelligence program. We found a compound that can directly bind to neuron and then destabilize this protein. So neuron won't be stable after we give the compound to the patients. And this compound can be easily taken up by the blood-brain barrier, can pass the blood-brain barrier, could be feasible for oral availability. So we have not given the compound to a real person, but I think it will be interesting to show you some movies of flies. So fruit flies are a very good model to study Parkinson's disease. I think I'll just give you some examples. So before we feed the compound to real humans, we can feed flies with this compound, and then we can generate Parkinson's flies. And those flies physically replicate Parkinson's pathology. So here to show you a wild type, let's say this is a healthy fly. And then you can see that here, this is fly, and then it just flies away because it is called fly, so they need to fly away once we release them. And then if we generate a Parkinson's fly, so this fly is we can express human alpha-synuclein protein that we just mentioned earlier in dopamine producing neurons in these flies. So we can make a Parkinson's fly, and you can see that it is very sad. This fly cannot fly. So the wings are still intact. The fly can still walk, but this fly cannot fly. So we can also examine local motor ability. So the typical cardinal phenotypes of these flies are the local motor defect and the death of dopamine producing neurons, exactly like Parkinson's patient. So it's a long movie. I'll show you. So it can jump a little bit, but it cannot fly. All right. So next, we could either give the compound, we can feed the compound to this Parkinson's fly, or we can genetically knock down neuron. So basically all these approaches, we reduce neuron in this fly, and then you can see it is like a miracle. So the fly just fly away, just like healthy fly in the beginning I showed you. So here, although they are not humans, but we have evolutionarily conserved the functions, and Parkinson in flies, and Parkinson's disease can be replicated in those flies. So it holds promise to show this compound could be also effective in humans someday. Okay, so lastly, we want to propose for future treatment strategy, we want to propose this precision neurology strategy for Parkinson's patients. So maybe Parkinson's disease, the term is already outdated because there are just so many molecular underlyings of Parkinson's patients, and the different disease may share the same molecular defect. And each individual patient is so different from others. So we propose, let's profile each individual patient. Let's do this genetic sequencing, looking at the demographics, neurological evaluation. Let's look at all the molecular defects we know, such as MIR1. You may know about GCAs and LRRK2 activity. So based on this profiling, we generate different strategies for different patients. For example, if this patient has both LRRK2 mutation and the MIR1 defect, we could combine LRRK2 inhibitor and the MIR1 reducer. And together with other symptom management strategies, such as deep brain stimulation or L-DOPA if the patient is still suitable. And then there's a cocktail treatment for each individual patient. And this cocktail would be different for each individual patients. So that's why we propose a personalized medicine strategy for future Parkinson's disease treatment. And again, we are at a very early stage, and I think people have put a lot of effort to develop this novel compound to correct the molecular defect. And we are very optimistic for the future treatment that we will be able to find a cure. We'll be able to have a cocktail treatment for every single Parkinson's patient. So here, I really want to thank my lab, who all these current and the former lab members who did the experiment, contribute to this talk, and the funding agencies and our collaborators. And thank you all for listening. I hope that I delivered the science that as clear as I hoped, and then you understand what I know, all the jargons I just said. And thank you for your attention. I'd like to take more questions.
Okay, yeah. If anybody has any questions, please put them into the Q&A or the chat box. And Xinnan, if you could go back to the previous slide.
This one?
Yes, that one. So one thing occurred to me about this slide is the blue figure. He doesn't have the MIR1 defect. Yeah. So I thought that everybody with PD had the MIR1 defect. So that's not the case.
So our data is 94% of the total PD patients show MIR1 defect. However, remember, there are also 10 to 20% of PD patients are misdiagnosed.
Right.
So it's possible that, that 8%, 6% in our cohort, they're not Parkinson's patients. It is possible, right? It's also possible that some PD patients just don't have the MIR1 marker, and then they are not, they don't need to take, they probably shouldn't take the MIR1 compound. It's entirely possible. I think the Parkinson's disease definition is solely both based on neurological evaluation. And given how we have known about the molecular defect now, we probably need to redefine this disease, maybe the MIR1 positive Parkinson's disease and then MIR1 negative Parkinson's disease, because the treatment will be totally different, right? So I think it's really traditional, like that's how we call Parkinson's disease, right?
Right. And that leads me to my other question, which is, you said when you first came to this slide that that the definition of Parkinson's disease seems to no longer apply, that our thinking has moved beyond that. Could you say a little bit more about that?
Yeah, so one example is that I just mentioned that Parkinson's disease patient may not have Parkinson's disease because of the misdiagnosis. Secondly, Parkinson's disease pathology share a lot of similarities. The molecular defect share a lot of similarities with other disease. For example, the GCA's activity is encoded by an enzyme called the GBA, and this GBA gene is also mutated in other lysosome storage diseases, a totally different disease, doesn't really affect the neurons, totally different metabolic disease. So then the question is why they have mutation in the same gene, right? Are there similar treatments for two very different diseases, right? They have very different symptoms. And then for LRRK2, for example, and alpha-synuclein, they are all discovered, the mutation of the same gene discovered in other diseases. Like alpha-synuclein is very interesting. Alpha-synuclein is even a pathology in, like, for example, Alzheimer's disease. MSA, we just mentioned, a lot of them have Lewy body formation, right? So then the question is, do this subset of the very different disease, they actually share the similar underlying pathological mechanism? If it's true, then they may share the same treatment strategy, right? So that's why I say that the term may not be, maybe outdated now, because first of all, the gene can be shared by other diseases. Second, the same disease may not have, they may not be the same disease. So I think from a scientist's point of view, it may be more accurate that we look at molecular defect, and then we go back to the biomarker question, right? So can we have a molecular marker that mark a subset of this disease, right? So they could share similar neurological symptoms. They may not, right? But because we know which protein is dysfunctional, then we can come up strategies to target that protein with a compound, with a drug. So that's what I meant when I came to this slide. Right. It's very complex and also very new emerging issue.
Yeah.
To a very old field.
Yeah, so I never took any of the sciences in college or in high school. I somehow got away with not doing that. And so when you say the word molecular, I'm a little unclear as to what that means. It seems that molecular can relate to genetics, so genes, and it seems that molecular can also relate to proteins. You said Nero1 was a protein, alpha-synuclein, of course, is a protein. Is that accurate, or what else would you include?
No, you're right. So molecular can be the gene, and the gene encode RNA, and the RNA encode protein, right? So if you have a mutation in the gene, you have a dysfunctional protein, right? So for example, LRRK2, molecularly, we can say the mutation is in the LRRK2 gene, then you get a dysfunctional LRRK2 protein, right? So it's a causal relation. And then that's how some of the PD patients may get the disease, because they have a dysfunctional LRRK2 protein due to the mutation in the LRRK2 gene. So it covers from the gene, the RNA, to the protein molecular.
Okay, good. All right. Somebody asks, I read somewhere researchers will find many types of Parkinson's disease, just as they've found many types of cancer. Is this a good analogy?
Yeah, I mean, really, it depends on how you look at it, because how you define types, right? You can define as genetically, like sporadic, familial, right? You can define as whether they're positive for Lewy bodies, and then you can define as early onset or late onset, right? You can also define that the Parkinson's patient with cognitive impairment or not. So it really depends on how you talk about it, because as I said here, each individual patient just has so many different parameters to them. The only thing they share is these maybe two cardinal motor deficits, and everything else will be different. Some patients have constipation, some patients have sleep disorders, some people have other dementia. And some people are early onset, some people are late onset. So I think it's just so complex, and you can define the disease in any angle you want. And that's why you can have a lot of types of Parkinson's disease. Yeah.
You mentioned that you have already tested one compound to deal with the MIRO1 defect. Mm-hmm. Is it just one that you've tested so far, or have you tested several?
So here, right? We tested. We also published the work. So at this stage, we tested at least 6 patients so far. And we're testing more now because we have the cells. We have about 100 patients cell lines.
And have you tested? How many of the MIRO1 reducers have you tested? These are the compounds. Have you just tested one, or have you tested several different compounds?
We tested about 6 patients.
Oh, 6. Okay. All right.
And we are testing more currently. Yeah.
Okay.
So at this stage, yeah.
Okay. It's 6 patients you've tested with one compound, or you've tested 6 different compounds?
With one compound.
With one compound. Okay. And if somebody asks, what is the realistic time frame for phase 1 trials? You knew this question would be coming, Xinnan.
Yeah, so I co-founded a company because this type of work is more feasible for a company to carry on, and they are working really hard, and they're also very ambitious. I believe the goal is hopefully sometime next year, we can submit an FDA application. So when that is approved, we can start phase 1 clinical trial, which is to only test the safety. And then we'll sure collaborate with movement disorder here at Stanford to enroll patients once it is approved by FDA. And then after that will be a larger scale phase 2 to test the efficacy. So I think in the next couple of years, hopefully, yeah.
Okay, good.
Work hard.
And if someone asks, does the MIRO1 defect also exist in vascular Parkinsonism? Do you know?
Yeah, I have no idea. Yeah. Okay. I think there are many, we could test that for sure. Yeah.
Yeah. Okay. I'll just read this question. It says that we have read widely. Some suggest that a leaky gut allows lectins to enter and reach the brain. A conference on keto reports research that ketones improve cognitive function. Are you able to comment on ketones or glucose metabolism, or is this out of your realm?
Yeah, I mean, I think, mitochondria are the hub for metabolism. So I think even CoQ10, right? A lot of people take that. I think any way to boost your mitochondrial function and the energy metabolism, like ketogenic diet, right? It could be very useful, and it may not. It could be useful for different people. Maybe very effective for certain patients, but not others, right? I think theoretically, these strategies is to improve your mitochondrial function. And as you can see that even our hypothesis suggests that mitochondria are not functional, right? So I think I don't know if that will really address the cause of neuron cell death, but I do believe, boosting mitochondrial function will be useful for certain patients. Yeah, that's my opinion on that. Yeah.
Right. Would you mind going back? I think it was one of the first slides about genes. It was a Medscape chart. And we've had some questions. Yeah, there we go. Maybe could you explain, I don't think you did, the difference between autosomal dominant and autosomal recessive. And related to that, one person says that you mentioned that Parkinson's disease can be hereditary. And what do you know about that? And if a parent has Parkinson's disease, what are the chances of a child having Parkinson's disease?
Yeah. So first start with autosomal dominant. So if you know the mutation LRRK2 or alpha-synuclein, those are autosomal dominant, which means we have 2 copies of our chromosome, right? Which means that as long as one chromosome has the mutation, you get the disease. And that is heritable, unfortunately. So if one parent has only 1 copy of their chromosome that has the mutation, it will pass down to your next generation. And then you can probably trace back to your previous generation as well. And then even in old days, they may not go to the hospital, but you may hear, Parkinson's-like symptoms, from your grandpa generation. So you kind of have some sort of family history before you, right? So this all information, this all important to confirm this is an autosomal dominant. There's an inheritance pattern, which means, so if you feel, fit in this description, it is highly likely your children are going to get a mutation. However, don't be too pessimistic. So there's another word called penetrance. So penetrance means that not 100% of the people who get this mutation actually get a disease. So there are many reasons that, factors could be environmental, could be nutrition, could be you have another mutation that contribute that you get a disease, but your children may not get. So for example, I believe mutations in LRRK2, only the penetrance is only 50%, if I'm not wrong. That means only 50% of people who inherited this mutation will get the disease. A couple very famous example, I should name the name here, that very famous people that we know in the Bay Area, right? They have the mutation. So those are also very late onset, right? And then given the improvement of our clinical management and also living standard, people just also get the disease later and later. So it's hard to know if, it's actually penetrant. You may never get the disease even if you have the mutation. On the other hand, mutations in this gene has a high penetrance. I think LRRK2 is only 10%, but this mutation called alpha-synuclein is 50%. So high chance if you do have the mutation in this gene, but it's very rare. This mutation is very rare. I think most people have the LRRK2 and another called GBA. So if you do have, your family has this history and then you have sequenced your genome, you do have this mutation, it is likely your children will, they will inherit your mutation. There is a chance they will get the disease as well. Yeah. On another hand, I forget what other question, but there's also a chance that if you just absolutely, your parents, your grandparents all sequence their gene in 23andMe, there's no mutation in LRRK2, but you somehow has it. So it's very likely it's a sporadic mutation, happen only to you. So that is hard to say because it's a sporadic case. So it may not pass, may or may not pass down because you will be surprised to know that 20% of normal population has mutation in LRRK2, but they don't have the Parkinson's disease. Yeah, so I think we'll need to combine your family history, the genetic trait of the gene, and then also what gene that you discovered in you, yeah, to make a conclusion.
And are these 11 the only 11 we know about?
No, sorry. So this is not updated. This is many years ago. Right now, there are like 20 or 30 new PD causing genes. Yeah.
Okay, so we can see that since this was done in 2006, more discoveries have been made for genes, and we can probably assume that more discoveries will continue to happen in the future of other genes.
Yeah.
Okay. And is it your hope with the MIRO1 reducer compound, is it your hope that, say, even if somebody has the first gene on this list, which is the alpha-synuclein gene, if somebody has that first genetic mutation, is it your hope that the compound will also help those people if they develop Parkinson's disease or even prevent Parkinson's disease?
Yeah, absolutely. So that's our hope. And then a very encouraging new study from our lab suggests that we can also screen at-risk individuals with this marker. So even before they get to the disease, we can discover them, we can spot this population, and then we can give the compound to them early on before they even develop the disease. So that will completely prevent the onset of the disease. And for people who already had the disease, they still have more than 50% of neurons alive. So if we can just stop there, and then, a lot of cases is their neurons are just dying, they're not completely dead. If we can clear out all their damaged mitochondria from their neurons can grow back, they can become alive again, because they're not completely dead. Once they're diagnosed, right? So I think our compound will do all this work, will clear out all the damaged mitochondria, make neurons healthier, they can grow back, they can become alive again, with the hope, like with patients even have already lost 50% of neurons, we can stop, even reverse the neurodegeneration process. That's our hope. Yeah.
Okay, wonderful. If there are any more questions, please type them in as quickly as you can. Otherwise, I think we've answered everybody's questions. Yeah, I don't see anything more in the chat or in the question and answer. So thank you so much, Xinnan, for explaining your cutting edge research and all the exciting things going on in your lab. And we certainly wish you the best of success and hope that this can get into at least phase 1 trials within the couple of years that you indicated. So thank you everyone for joining us, and thank you again, Xinnan, and we'll see everyone next time. And Xinnan, if you could hear everyone clapping on Zoom, you would certainly have a loud applause. So
thank you so much, everyone.
Thank you so much. See you everyone. Have a nice evening. Bye-bye.
Bye.